So we'd like to start something else before pharmaceuticals perspective evening sessions. We realized that there are a few things still needs to be filled, something outstanding or additional section may be needed. So Merit raised some efficiency of clinical trials, and also Ben Brooks talked about verbal and cognitive outcomes, and also Dara mentioned how we can tell the patient when we found a genetic mutation in the study through clinical trials. Those has to be added into clinical trials, clinical trial guidelines through recommendations. So maybe Merit, would you like to mention about this?
Oh, sure. This came up when we were talking about whether we could be faster by combining phases of trials. And really, a lot of the delay in getting to answers isn't so much about combining phases of trials. It's really about getting studies off the ground once you've designed the study and funded them. So ideas like being promoted by the NIH of using a central IRB and master contracts. So we know from doing this in NeuroNext that instead of a year from the time you're funded to getting your first patient in, using a central IRB and master contracts can get that study startup down to 30 days. So promoting something like that in ALS, where we use a central IRB and we use some standard contract language, could greatly accelerate trials, and I think would be more effective actually than combining phases of trials. And then related to that would be a lot of the delays to getting answers is that it takes so long to recruit trials. So working on ways to recruit faster would go a long way.
Is the does NEWS have such central IRB? I thought I heard that process.
Yeah, so the ALS Association has given some infrastructure funds to NEALS to create an ALS central IRB. We're launching it on a biomarker study as a kind of a pilot with that James Berry is leading, but we'd like to pilot it on one of the new phase 2 trials that the ALS Association and ALS Finding a Cure are going to fund.
Certainly, this should be included into this guidelines and recommendations.
I think so.
And I don't know who is going to take this very important topics, because we have to move clinical trials fast, facilitated. So anyone volunteer, that would be great. If not, we're going to assign, and so to do this process.
I'm happy to do that. Maybe
Okay, someone voted Merit, so.
That's what you get for having speaking up in this crowd.
All right, great. So next, Ben, would you like to stand up and discuss this verbal and cognitive outcomes?
Actually, this is a little easy because Eric Macklin today recommended that the NIN-CDS common data elements be the components for doing the clinical trials. And all of the scales that we've talked about are there, I checked. And so where we haven't really covered in terms of bulbar scales are in there with some of the modern scales. In addition, also all the cognitive scales are in there. So I think if we just make the reference that Eric made, then we'll be able to cover in the guidelines.
So do you suggest that Eric to add those sections?
Well, no, I'll revise the sections that are in the guidelines based on what his recommendation was.
Excellent. Okay, good. All right, and then last things is, yes, Dara, would you make comment about the genetic mutations?
So I was asked to do supplements before, so I keep getting these controversial subjects to at least discuss. And if you'll see tomorrow morning, we've, in our section, completely gone the other, given the other message about what to do for supplements and trials. So I emailed Hiroshi earlier. I said, what about the issue of genetics and finding out about genes during the course of a trial? So we're currently participating in the ALS Cosmos study. And the samples that we were taking over the past 5 to 8 years have been looked at again, and then we found C9orf. What do we do to tell these patients or their families, many of whom, most of whom have passed away, they're C9orf positive? So it's a significant ethical problem and controversy, but then it's also looking at the future. What do we do when we do these studies, or looking at all these biobanks? If we're going to do genetic testing, which has been promoted here, that we should do DNA screening, how do we deal with patients and their children? This is I think, an inflammatory, very sensitive issue. I think we have to be very pragmatic how we approach that. So I don't have a good answer for this. And Michael Benatar, Michael, are you here? Because you've been doing the familial ALS. I guess he's not here, but I don't know who has been doing a lot of the consults and dealing with IRB. I don't have the best answer for dealing with genetic screening. And I don't know, Tipu, you may.
The ethical requirements are that if you come across an incidental finding that is important to the health of the patient, it has to be disclosed. The second is that it has to be part of your IRB when you undertake genetic testing as to what your methodology is going to be. If it is known to you, in other words, the identity is known, then you have to, in the informed consent, let it be known whether that information will be disclosed or not disclosed. And this has been also studied in England about the disclosure issue. We have always taken this position that we will disclose if requested, and because there's no treatment. If there is treatment, then you are required to disclose.
So the IRB, the consent form that we did was not to disclose. So in my heart, I feel that's wrong. So I mean, that's one thing. There's law and heart about what's right and wrong. But number two, we will have treatments in the future, and that's where it's. I'm being optimistic. We're going to have some.
I think the issue there is that if you have treatments, then you have an open-ended agenda in which you communicate with people and ask if they are interested in knowing, not to tell them, but if they are interested in knowing. And if they are interested in knowing, then the disclosure is made through proper genetic counseling, just the way it has been set up for Huntington's disease. We have set that up, at least in our center, where a genetic counselor then counsels the patient, has several sessions, and then the information is disclosed. But you have to sort of be open-ended about it rather than say, I'm going to tell you.
So I think it's very important to think about this ahead, because the first thing to say is if you think 5 to 10 years ahead, across the world, it's going to be the case that most people will be able to access their genome readout. Okay, so this is a sort of transient period of history when we're struggling with this morally as to what you should do with information that you have, that the patient doesn't have. That's going to change completely within 10 years, okay? But for the next 10 years, I mean, what matters is what the patient's actually consented for. So that's, in our system in the UK, certainly, it's not, we don't start sort of in a post hoc way wondering whether we should disclose information. It's actually what did that person, when they entered our study, sign up for. That's really what you have to be very clearly guided for. It's pointless getting into the situation where you know, in a post hoc sense, you have some information and then you're struggling to know whether you should divulge that. I think when someone's died, if their family come along and say, my father died of ALS, what do I want to know more about my own risk. That's a completely different matter. That's a clinical issue which you can then address, in a different way. So I think it's quite clear that it's about counselling people at the outset of their entry into the study, but not getting into kind of complexities later on when you feel, you're in possession of information that you don't know how to divulge. What has that person consented for, is the key thing.
Do you think you can take this in your sections to written recommendations?
Right. So it's difficult because we haven't really included it, I think, in any of the. It's sort of a new area, isn't it? So we're going to have to sort of start from scratch, really.
We'd like to have someone to take this important topics.
I mean, I don't know if Adriano Chio is here, but he's written extensively about this. Adriano, do you want to say something? Because I mean, you've actually produced a sort of.
So you want to take?
We had a consensus meeting in Italy to discuss about the use of genetic testing, and we concluded that if there is familiarity, genetic testing should be done and should be given to the patient unless the patient call out. But the problem is for sporadic patients, because a lot of them, not most, are really genetics. So this is a point that we think that genetics should be done in anyway. So this is a point, in our opinion, in Italy, we do that now, and I think that also Vincenzo does the same. And if the patients do not want to have genetic, okay, but we propose them to have, and this is our position.
So why don't we have one more comment, then we're going to have to.
Can I just say one practical solution to the ethical problems that you raise is, though the IRBs and our consent forms for patients said we do not consent to being reapproached after the end of the study, one of the possibilities is social media and all the other media to release the information that we have such information for those who took part in this trial if you wish to contact us. Yeah. So that's a possible way around the ethical dilemma that we're in.
If you ask at the moment of when you obtain the blood, and you can ask the patient if he or she wants to have the information, so you are going to give it. If he or she doesn't want, well, there is no reason why to reveal what's the result. But it is important for the trial.
I don't think we need to answer at this point. We have to produce recommendation from us.
Well, it's a really important and very passionate question. In fact, at the recent NEALS meeting, we had a whole symposium about this issue. Chris Shaw has recently told us that he believes that, in fact, the C9 mutation is not very penetrant based on his data. So I don't think we can make this decision now. I think it requires a lot of conversation by a lot of experts and ethicists as well. So we need to move on now, if we could, to the evening session. And the evening session is about industries, perceptions probably, lessons learned from future drug development. And the first speaker is going to be Don Johns from Biogen. Don is the Vice President for Clinical Development. And he's going to, the way we're going to do this is each speaker is going to speak for about 10 minutes. We'll take just a few questions between each speaker, and then we'll bring all the speakers back at the end for kind of a panel conversation. So if you don't get a chance to ask a question right away, there will be time at the end to ask questions. Don?
So thanks, Jonathan. I actually want to thank Hiroshi for inviting me. So I'm sort of hoisted on my own petard, because when we were discussing this, I suggested we have this session, and then he took that and then said, good, you'll be the first speaker. So I be careful what you wish for. So I'm actually, for my disclosures, I'm a full-time employee of Biogen. I have stock in it. I'm so conflicted that it's just overt. So I don't get money from anybody else. And so, but at least mine is declared. So I think, and actually I'm going to give a Biogen, I think each of us are going to give somewhat of an idiosyncratic perspective of how we deal with things. I think Biogen has a very integrated approach, and I'll outline that for you. There are many good ways to do this, but this is the way my unit is currently doing it. So we're actually sort of integrated fully to discover and develop treatments for ALS. And obviously the strong motivation, the profound unmet medical need is so apparent. I just want to mention the clinical know-how as demonstrated in the operational performance of the EMPOWER study And just on an anecdotal basis, one reason I came to Biogen after a career at Novartis and an academic career prior to that was, despite the fact of a very expensive failure of the phase 3 study, Biogen actually chose to double down on ALS. We recruited Chris Henderson, my colleague on the research side, who, as many of you know, is one of the world-class researchers. And the institution was so touched by both the profound unmet medical need as well as the emerging science that they said, no, we're not walking away from this, we're going to double down. Very few companies, particularly big companies, would have done that. It was very motivating for me to come over. And I mentioned Chris Henderson, who's an important part of our emerging science, that we have obviously human genetic advances, a rich supply of targets, and a multiplicity of ALS models for which we do in an integrated fashion. In addition, on the Empower study, it's been a very fertile source for looking at the potential development of novel endpoints and to mine that data very thoroughly. So I'm just going to mention this is bringing coals to Newcastle to tell people about the profound unmet medical need, but there's a couple points I'd like to make here. One is that we really need to think of ALS much more like cancer. It is similar to deadly forms of cancer, such as ovarian and lung cancer. If you look at the 5-year survival is very similar to ovarian cancer. It's not another neurodegenerative disease. It's really, it needs to be thought of very differently. Just another couple of points is the incidence is almost half of that of multiple sclerosis. It's just in my company, multiple sclerosis is a pretty important disease, and people are a little surprised by that. And finally, just a publication that's actually been cited a lot at this meeting, with that more than 50 failed ALS RCT trials, and that I think that's really some of our profound motivation for many of the things that we've been discussing here in this venue. So just a little bit about how we've set things up. Obviously, we've gone over a lot of this in this meeting, reasons for prior study failures, targets unknown or unvalidated, heterogeneous patient populations, lack of mechanistic biomarkers, grossly underpowered phase 2 studies, particularly for efficacy endpoints, and insensitive measures for functional rating scales and death that really are insensitive to short-term and small n changes. So what we've tried to do is to pursue validated, genetically identified targets initially. We'll expand the target from there, and to data mine the Empower study to identify some potential novel endpoints to help answer many of the questions that perturb us now in ALS clinical trial development. We're doing a defined methodology study to help validate endpoints and to do side-by-side comparison in a longitudinal manner. So the ALS genetics have really, I think, has been probably the most profound scientific change in the last decade, and really is guiding both target selection and clinical study populations. And I just want to highlight one of our own internal efforts, that was the Exome sequencing study, which identified a novel gene for ALS, a TBK binding kinase, and that really implicates strongly the autophagy pathway, since its substrate is optineurin and known, and also P62. So it really points out, in addition to the small number of patients that have this mutation as a causation, that the autophagy pathway may be a fertile area to look at for future drug discovery. So our rational first target for ALS is SOD1, and I think the reasons are quite apparent to all of you. The first identified target, very robust preclinical data. I think most people you may not like the SOD1 mouse model, but I think for a SOD1 directed therapy, even the most skeptical people will probably give you a pass on that. I won't say it has a lot of generalizability beyond that, but in this case, it actually we have quite robust data. So we're actually working with Ionis Pharmaceuticals, the artist formerly known as ISIS Pharmaceuticals. There's some very interesting stories about that, including someone waiting with a sign in an airport that said ISIS Pharmaceuticals to their driver was treated rather rudely. So they eventually changed their name after they appeared on Saturday Night Live. I don't know if you guys saw that. They were lampooned on Saturday Night Live. On Monday morning, they changed their name. But they're very good partners, Ionis now. So we can look at a genetically homogeneous population, although there are a number of different mutations, and we have a very robust mechanistic biomarker, which is the decrease in SOD1 levels in the cerebrospinal fluid. and this is really based on the former SOD1 study that several of you in the room participated in, that show the stability of intra-patient stability of CSF SOD1 over time, which is a key mechanistic biomarker. So we really need better endpoints, and I think that's been a sub-theme here, something that is sensitive and specific to track disease progression, something that can detect changes in a shorter time frame, require fewer patients, however is clinically meaningful, and more importantly, has forward positive predictive value for the currently FDA-accepted registration endpoints of ALS, FRS, and survival. Also, these markers have to have robust operational characteristics, being objective, standardized, and feasible, reliable, and reproducible. A high bar, but one that we have to really aspire toward. And so I mentioned our methodology study, which actually prior to the Ice Bucket Challenge, we may have had the only methodology study going, but now after the Ice Bucket, Lucy, there are lots, I'm glad to see there's now many efforts for methodologic and longitudinal studies. But we really want to do a longitudinal head-to-head comparison of a number of measures, really to, I think that the weak point in ALS drug development is really the phase 2 proof of concept level. Phase 1 you can do fine. Phase 3 you can actually, we can recruit and crank through. This is the weak spot. So we've really I've focused my group's efforts on the sort of proof of concept phase 2 area. So we're looking at strength measures, electrophysiologic measures. It's one of the few studies to actually do all of these electrophysiologic measures in the same patients longitudinally. You do a lot of individual technique and then compare across studies. This will be within the same study. We're also doing spinal cord imaging because there's a remarkable dearth of spinal cord imaging data. There's a lot of cortical imaging data. Martin Turner, many people in the room have generated a lot of that. We chose to actually fill a bit of a niche. Really the French group is about the only one that's produced much of this spinal cord imaging data. And we're trying to do this longitudinally. So just a future view of ALS clinical trials, and just sort of the previous clinical trials that took all comers, really to be fully powered require about 220 patients per arm for a modest change using the outcome measures of ALS, FRS, or survival. Treatment period was 12 to 18 months. Biomarkers have rarely been done. The cost is, for comparison purposes, 5x. And there's just a remarkable number of these that have failed, many of the vast majority of which have been underpowered phase 2 studies that purported to look at efficacy but were grossly underpowered to do so. So what we're working on is an approved proof of concept approach applied to both familial and sporadic ALS, with the patients really depending on the characteristics of the therapy being tested. We're aiming to have a much smaller N, and we're using a novel measure that we're in the process of validating and developing that allow us to, in 6 months with a smaller N, determine a robust and clinically meaningful effect. You need to have biomarkers of engagement. It's been talked about in this study today, and actually at a much lower cost. It increases your aperture to really test more potential therapeutics. So finally, just to finish with what I think is an integrated approach, really aided by genetics, we're trying to define ALS in actionable molecular pathophysiologic terms, validated targets, eventually in causal biological pathways for low molecular weight development. For now, we're starting with a new therapeutic approach to accelerate gene to drug, and that's ASO. It's remarkable how you time compress the process by years to have, from an ASO, from target selection to therapy, is radically faster than a low molecular weight approach where you spend years going through medicinal chemistry, et cetera. I think perhaps the most important point I'd like to make here is that we see at Biogen, we want to contribute to the ecosystem of ALS therapy discovery and development. And we're working with many of you in a variety of ways to sponsor things, fellowships, et cetera, to sponsor natural history studies, et cetera. And we really see our roles as really part of the ecosystem. It's a healthy ecosystem that requires a lot of different people. I was an academic for 25 years and I haven't forgotten most of that stuff. So hopefully I can actually bridge between those a bit. We really take an integrated research and development effort and really focusing on efficient early phase studies because it's the gateway to phase 3. The Empower study, was recruited very quickly and robustly. We've got that covered if we get something there, but I wanted to work in this space, have validated targets with target engagement biomarkers, defined ALS subpopulations, stratify that sub-study population by key parameters. We're data mining the former Empower study to help us do these things, and we will eventually have robust, sensitive measures of disease progression that are sensitive more than specific. In this phase, you don't want to leave you want to avoid a false negative. There's only one approved therapy in the West. We don't want to leave a potential therapy on the table. And finally, just the sort of bottom line of all this, our SOD1 ALS phase 1 study started in January of this year and is proceeding well. Thank you very much.
Any questions for Dr. Johns? Leonard?
Can't hear you.
Thanks, Brian. I think the lifetime risk of MS and ALS is about similar. So you could get more money out of your company to say that to your management.
Yeah, actually, they seem impressed by this figure. I'm going to stick with that for now. That
similar is twice as much as half.
We still have most favored nation status there. I'm not going to jeopardize that.
Okay, well, we can bring in some people too. Amar, it's the same, is it? It's the same. So.
Yeah, it's just remarkable when you look at the incidence versus the prevalence because of the duration of the disorders. But it's an interesting figure. People think of them as being radically different in their frequency, and they're really not when you take into account the duration of the disease.
Yeah. The lifetime risk of MS is almost exactly the same as ALS, but the sex ratio is reversed.
Right.
And the real difference is the distribution across the years.
That's an excellent point. Yeah, the longevity.
All right. One question about the data mining that's going on for Empower. Can you talk about the actual data that you're looking at? Does it include molecular level analytes from core samples from those patients' blood or any others?
So some of that's being generated. A lot of that wasn't actually generated at the time, but the samples have been stored to do that. We focus most of our efforts on the clinical data because there's a wealth of data. This is remarkable. Since it was a phase 3 study, the data is pristine. It's been source verified, et cetera. Most natural history studies just don't have the same really quality of data, even though they can be very good studies. Since this was meant for a registration study, the data quality is really remarkable. And to have an N that big is also quite remarkable.
But as far as the assays and genomic characterization of those patients at baseline, not much was done, but you've still got the samples, is that right?
That's correct. Actually, there were some issues with the way the genomic samples were actually done. When they were done in the course of the trial, it caused a little bit of an issue with that. But we do have biosamples available from them that we still sort of, are now planning which are the best assays to do and when. My red light is going.
So I was curious. ASO means antisense nucleotide. So every time you do a clinical trial, do you have to do a phase 1? Or since it's always basically the same compound, antisense nucleotide,
No,
the target?
You have to. It's not the same compound. That would be like saying every low molecular weight compound's the same because they're all a pill. They're absolutely each unique and have to go through the same rigorous process, even actually all the steps, because they're really unique molecules. So interesting enough, within the FDA, ASOs are considered like low molecular weight compounds. They are not considered biologics. So they're actually classified under the CEDAR, you remember the guy from the FDA, all the way to the normal drug pathway. Now, we have to do a lot of education. I think when we went to the EU, there's a lot of education. People haven't seen ASOs before. And when someone's not familiar with something, I think we all regress back to a more conservative approach, right? So we've actually been fortunate to have pre-IND meetings with the FDA and have good discussions. And they've seen ASOs before, several of them actually now, including the first SOD1 compound, but our SMA compound and some others. So the familiarity of people helps them to know some comfort level about what are the key questions to ask. Thank you.
Thank you, John. So our next speaker is Dr. Andrew Wolff from Cytokinetics.
So thank you. I'm going to address a topic that has come up a few times now during the meeting, but I don't think has been discussed in much detail. And that's whether or not we should view mortality, or even mortality and morbidity, as really the gold standard for approving new treatments for ALS. I recognize that there are other endpoints that are deemed acceptable, like the ALS Functional Rating Scale, but this one comes up a lot. And I'd like to really examine this in some detail. Now, first, I want to make just a couple of disclaimers. One, these are my personal opinions. I'm not speaking here tonight on behalf of Cytokinetics. I didn't include the corporate name or my title on the first slide. There's no logo. This is just me talking to you. But that said, I think it's valid to include it in an industry perspective session because they are my personal perspectives based on nearly 3 decades of doing sponsored research for industry and also the regulatory interactions that accompany them. So I'm going to start with a proposal, a statement that may seem provocative to some. I don't intend for it to be, but I just expect it will be. And that is, we don't help ourselves or people with ALS when we refer to high mortality in ALS. And I'll explain why, and in some detail over the next couple of slides, but in summary, as I'll show you, mortality per se in ALS is really not that high compared to other more common diseases. It's really the incredible burden of functional loss that distinguishes ALS, not so much that the mortality rate is so high, but more importantly, when we refer to high mortality in ALS, we hand an argument to regulators and others who tell us that because the mortality rate in ALS is high, we should be able to easily execute mortality studies in ALS. And if you hold that view, I'll tell you right now, I hope to disabuse you of it over the next few minutes. And further, because of this, I would propose that we eliminate any references to high mortality in the 2 documents on which most of us are working, the clinical trial guidelines and the draft FDA guidance. So let me show you why I'm taking this position. So let's compare mortality in ALS with mortality in acutely decompensated heart failure. I don't need to tell you about Empower. You all know about Empower. Everest, importantly, was a study in patients who had been admitted with acutely decompensated heart failure and then were stabilized, and within 48 hours of their admission, they were randomized to an arginine vasopressin antagonist, tolvaptan, or a placebo. So these are patients in the hospital after an acute decompensation. So let's look. Here's the mortality, all-cause mortality in Empower, 18% at 1 year, 24% at 18 months. We're all familiar with that. Here's all-cause mortality in Everest. It's actually about 50% higher than all-cause mortality in Empower. But, as I mentioned earlier, clearly, one of the major differences is these heart failure patients feed themselves, they dress themselves, they bathe themselves. They are essentially all ambulatory, and certainly, in comparison to patients with ALS, they carry very little functional burden until the day that they drop dead from a ventricular arrhythmia, which is the leading cause of death in these patients. So the mortality isn't uniquely excessive. It's too high. I understand that. I'd never argue otherwise. But it's not the mortality that's unique to ALS. So what do these mortality rates mean? If we take the position that you need to demonstrate a decrease in mortality as a gold standard for approval of a new treatment. So again, the most recent study, large enough and long enough to give us a reliable point estimate of mortality in ALS, was Empower. It was 18% in a year, and we want to do a phase 3 study, so we want 90% power. We need a two-tailed alpha of 0.05. Here are the numbers. Thousands of patients. taking 3.5 to 4.5 years, and that's just to get the last patient visit. We still have to get the data in, we have to clean it up, we have to lock it, analyze it, put it into an NDA, get it into regulatory authorities, and according to conventional regulatory advice, unless the p-value is very much less than 0.05, you need to do another one. One won't suffice. So if you're a very large pharma company with very deep pockets and an extremely high level of confidence in your treatment, maybe you'll run them in parallel, but probably not. So that means the timeline to an approval based on this method, this approach, is upwards of 10 years. And clearly that's not acceptable to patients with ALS. It's not acceptable to us, I'm sure of it. So what can we do? Well, we can use a composite endpoint. That'll get us some more events, and that'll increase our statistical power, and we'll need fewer patients, and we can do it faster. And one that we know regulators will accept is time to death or respiratory insufficiency, which was studied in Empower, and it had a very strict definition for respiratory insufficiency. And what did that do to the event rate? Not much. 3 percentage points difference between the composite endpoint and just all-cause mortality. So that didn't really move the needle. Well, what about another composite endpoint? Another one that's reported in the Empower manuscript is time to death or a decline in the slow vital capacity to less than 50% predicted. Well, now that did really move the needle. That essentially doubled the event rate. And so, if we could use that as a primary endpoint for clinical trials, that would somewhat reduce the number of patients we would need to enroll and the time it would take. But regulators do not view measures of respiratory function as reflecting clinical benefit as they view the term. And so a composite endpoint that includes such a measure is not acceptable in their view as demonstrating clinical benefit. And I have to tell you, even if it did, it doesn't really solve everything. So let's look at what the numbers are if we could use that composite endpoint compared to all-cause mortality. It's still almost 2,000 patients. It still takes almost 3.5 years. And even if we had an even larger treatment effect, what if we could reduce the rate of that composite endpoint by 25%? It's still over 1,000 patients, and it still takes 3 years to do. So I'm suggesting to you that these sorts of studies can't be the gold standard. They're not feasible. And we do have other proposals, and I understand that. I just want to argue that this is not the way forward. So here's where we are now. I think I've shown you that all-cause mortality as an endpoint for an ALS clinical trial takes thousands of patients, requires several years. Even if we can use a composite endpoint, we can reduce the number of patients by about 1,000. That's not trivial. We can shorten the study by about a year. Also not trivial, but there's still thousands of patients in several years. In contrast, a study that is very well powered to demonstrate a difference between placebo and active treatment in a continuous, reproducible, objective measure of respiratory function, like vital capacity, but not necessarily vital capacity, could be SNP, could be something else. That could be done with hundreds of patients, and it can be done in a year or two. But currently, regulators won't accept it as a primary endpoint. So what can we do? Well, one thing I propose we can do is to write these clinical trial guidelines that we're here to do, to say that objective and reproducible continuous measures of pulmonary function are measures of patient function. And furthermore, they're measures of the patient function that is most likely to determine their eventual outcome. And so they do reflect clinical benefit. And I'm proposing that we be very clear about that in these guidelines. And furthermore, I'm proposing that we be clear that a positive impact on such measures should be able to be the basis for approval in a phase 3 study. But I live in the real world, and I recognize that even if these guidelines do say that, and I have no idea yet that they will, I know that the Outcome Measures Committee committed to give it a thorough evaluation, but I don't know what they decided. We'll find out tomorrow. Even if they do, it may not move regulators from their current position. So what else could we do that actually might work? A novel endpoint that I don't believe has ever been studied in a clinical trial, but that we have reason to believe would be acceptable to regulatory authorities, is a composite endpoint of time to death or initiation of assisted ventilation, mechanical ventilation, whatever you want to say. I don't want to say NIV because it should include everything, but we don't have a standard definition for what is initiation of assisted ventilation. Is it when the investigator prescribes it? Is it when the patient first uses it? What if the patient uses it for one night and says, that sucks, I'm not doing that again, and we all know that happens. Is that an endpoint? Or do we need evidence that the use of assisted ventilation has been more sustained? I don't know. You're the experts, but what I'm asking for is a definition that we can use. And in addition, we all know that treatment guidelines and practice patterns about when to initiate NIV are widely variable across regions and even between centers within regions. That's another problem that's not within the scope of this group to address. As we've explored this, people have said, well, a decline to less than or equal to 50% in percent predicted SVC is a pretty good surrogate for starting non-invasive ventilation because that's what a lot of people do, but we know that not all people do that. There's great heterogeneity in here. So I'm proposing we need a clear and widely accepted definition for what constitutes initiation of assisted ventilation and also consistent standards for when it's instituted. That would allow us to do clinical trials that are somewhat smaller and faster using a primary endpoint that we can be fairly confident could support approval, and that would allow us to accumulate endpoints faster and more predictably so that we could design and manage clinical trials in ALS more predictably. So in summary, I have a few final thoughts. One, I'd like to suggest that when we make references to mortality in ALS in the two documents that we're all engaged on right now, the guidelines and the guidance, we should reference objective measures of mortality based on data that has been recently produced. And we should not use qualitative terms such as high, because it only feeds the view that the mortality is high, and so mortality trials are feasible, which I hope I've convinced you really they're not. I understand that treatment standards that can harmonize when NIV is initiated are not within the scope of this group, probably nor within the draft guidelines group or guidance group, I should say. But a definition of initiation of assisted ventilation is well within a clinical trials guideline working group. I know it's the outcome measures group, and so I respectfully request and strongly urge you to include that as a potential phase 3 endpoint, death or initiation of assisted ventilation, and please, a definition for what constitutes initiation of assisted ventilation. Thank you.
We have time for 1 or 2 questions.
You get an A for trying, I must say. And I'm with you. I'm with you in trying to do something that's better than what we have. But I guess it diminishes the problem. And the reason I say that, from what I remember, class 4 heart failure is bedbound. Class 3 is barely able to walk.
No, that's actually not the case, but okay.
Yeah, 4 is bedbound for sure.
4 is symptoms at rest. 3 is symptoms with moderate exertion.
So that's sort of end stage of ALS, to be bedbound. And we're not looking at the initiation of disease in that New York heart failure 3 and 4. We are looking at the end stage, and it's exquisitely age dependent. For example, if you're 80 years old and you have heart failure, you're going to die in 2 years, as opposed to if you're 50 years old. So there's a big difference of age, that age dependent heart failure, death, mortality. So that's the first issue. So I think that analogy it will sound weak to people outside this room. We would go with you, but I think outside. The second issue is that I think we can make a case that we want to see the effect of a drug or an existing drug on forced vital capacity, or SVC, not as a treatment measure, but on the effect on a parameter like you would look, for example, on sodium or potassium, without saying that this is a treatment for the disease. And I think that may get your foot in, and then subsequently that would possibly lead to a change in the heart of the regulator.
So I'm not sure I agree with your points about heart failure. I think they're exaggerated, but that's okay. It's not really the issue. The issue is that as high as the mortality in ALS is, it's still not so high that you can run a fairly small and short clinical trial. It takes thousands of patients in several years. That's my point.
That's a major
Andy, Jess Rayborn, I thought your talk and the points in them were very clear and compelling. You know probably what I'm going to say, and that is that WIDE Trial is very interested in helping you running a central protocol multi-site access trial where we might be able to help validate this new composite endpoint. And we'll pay for it.
So let's talk about it.
I'll leave it at that.
Sure.
Very nicely argued, and we agree wholeheartedly. The Neil's Ventilator Subcommittee, chaired by Christopher Lee at Vanderbilt, I think is going to be a place where some of these things should be worked out, in particular the time to failure for NIV. We use that endpoint for our Ibutalast trial, and we'll be using that as well. We haven't looked at the composite endpoint, and that's a very interesting thing. But and maybe the statisticians can help us out. I mean, the O'Brien-Fleming analysis of more than one multiple endpoint. Have you looked beyond the two that you've looked at yet?
No, not rigorously. I looked at that one because the data were easily accessible in a recent clinical trial.
Speaking as a...
That's fine. That's fine. And just if you could make them short questions.
Okay.
Sure. I think we understood from your talk that you'd need 1,200 individuals for this composite endpoint of mortality or SVC below 50%. But you only need 400 or so for a trial tracking ALS FRSR over a shorter interval. Does including this endpoint really bias something?
Well, I don't know that ALS FRSR moves very much over a shorter interval. That's one of the things. And I wanted to be clear at the beginning that I recognize ALS FRSR is deemed acceptable and could be used, but I wanted to focus on what I hear, that mortality should be the gold standard, and that I directed my remarks there.
Yeah, I was just going to echo what Eric said. Smaller sample size, because when you take a time to an event, only the events count towards power. When you use a continuous variable like rate of decline, everybody contributes to
I'm sorry,
power.
Continuous variable like what?
And it's going to be, well, like he said, to be 400 if you're looking for a 25%.
No, yeah, I know. I'll just say that ALS FRSR is not a continuous variable.
Well, for FVC or SVC, it would be similar. There's greater variability, so it'd be it might be 25% higher. But it would still be less than a time to event study.
Okay. I was here. Go ahead.
So we're going to have some more time at the end. So thanks very much. Next is a presentation from Knopp Pharmaceuticals, and that's Michael Bozik.
So appreciate Hiroshi's invitation to speak on industry perspectives. I sort of feel like I appreciate the invitation in particular because we been sort of relegated to the bench for the last couple of years, and actually are doing some different things at Knopp Biosciences. as it relates to Dexpram and Paxil. But I wanted to talk about something that I think is really interesting, which is, when do you stop when do you stop looking? And I put this slide up, and pardon the parochialism of using an American football analogy, but I think the use of perivascular tau tangles, neurofibrillary tangles, sort of unifies us all. But I think everyone's a little bit familiar with the story of chronic traumatic encephalopathy. For those of you who haven't seen the movie Concussion, there's a hero to the movie. His name is Bennett Amalu. He's a Nigerian neuropathologist who did an autopsy on this fellow who I think Stacy may be the only other native Pittsburgher here, but Mike Webster was an all-pro Steeler who died homeless under a bridge after lots of self-inflicted injuries, and was taken to the corner. They were ready just to sign off on him, and this fellow thought, there's more to see here, and he pursued it. And this fellow, who might represent the industry, didn't think it was such a great idea. In fact, when his paper was published, Roger Goodell and the NFL tried to get the Journal of Neurosurgery to retract it. Interestingly, this is now accepted as the definitive, the pathognomonic lesion for chronic traumatic encephalopathy. And ironically, on March 15th, 3 days ago, the NFL finally acknowledged in front of a House subcommittee that chronic traumatic encephalopathy clearly is a result of repetitive concussions in football. So, when to stop looking? There are different perspectives on this. And the other thing is, I want to say, when you ask for industry, and I heard some interesting comments at dinner about, we're just going to be preached to by the industry, I think was the comment, something like that. The industry is about as heterogeneous as is ALS. You have here tonight a large cap company, publicly traded, sales of over $10 billion last year for the first time in their history. Congratulations, Don. I know, but those stock options are looking good. No, just kidding. No, I mean, but very different business than Cytokinetics, small cap company, pre-revenue that's been really slugging away and took a different view about when to stop looking. It's been around since 1997 and has continued to push. And then there's what I'd call with Bernard a new cap company. Just went public last fall. Whether it actually got out at a time when nobody thought they could get out, so good for you, Bernard, and the Voyager team. And you have Knopp Biosciences, which is really a privately held company that sort of is funded with or held together with rubber bands and chewing gum at this point. So, like Andrew, I'm going to offer my perspectives. They're pretty much Knopp's perspectives because we don't really distinguish a lot. But so I'm just going to share with you a few thoughts. Let's see. Did I turn it off? Where's my oh, there we go. Okay. So this is a study that everybody's familiar with. Pretty crappy outcome. And I think it's fair to say that there was just enormous disappointment, maybe the most disappointing ALS clinical trial ever. So do you look at, do you look at all? Okay, the body, it's a patient's dead. Well, you do. And Biogen did. And this is what Doug Kerr said at the time. In fact, interestingly, if you want to hear what he said, the ALS podcast is still available to you on the ALSA website. He said, we looked. The first thing you can't, what if the trials were different? It's like, well, they weren't. They were identical in every respect. They were identical populations. And as a consequence, that was where they stopped looking. And George Scangos not only said, we're going to stop looking, but he published an op-ed piece in Business Week and said, you shouldn't be taking this drug. Not only don't look anymore, but don't ever take this drug. We have a responsibility to the patients. Turns out the study populations were not exactly identical, and in fact, there were some very significant differences in symptom duration, riluzole use, and maybe most importantly, the ALS score definite criteria. And the reason this is circled is because the next two slides are going to, or the two slides after this are going to get at some data that John Katz and Dan Moore looked at and shared with me, and I appreciate that. But we heard a lot about time to enrollment. Well, you know, this was, let's see. This is the kinetics of the Empower enrollment. And, you know, if you look, what you'll see is there were about 140 patients enrolled over the first 4 months. Over the last 2 months, there were 600 patients enrolled. Now, and I also heard some regulatory disparaging last night about, you know, validating and checking things. And what's interesting is if you go back and look, not only do you have this depletion of ALS definite patients, there were patients, not many, but 5 in that period of time that didn't even have ALS. And there were certain entrance requirements, especially 2-year requirements, that a different analysis, and this is not an industry analysis, this is a Forbes-Norris PROACT analysis that looked at, well, can we use historical, you know, how steady is the ALS FRS? And I think it is steady. If you look at population data, and Dan can correct me, but I think LOES stands for locally weighted smoothing, scatter plot smoothing. Yeah. So, and basically, I think that's a fancy word for a regression analysis. But what you can see is there was very tight. These were the authors' conclusions, and they said, you know, if you look at the minocycline study and the Teva study of telampinel, very tight, very predictable. But what did they find when they looked at the Empower study? Well, they actually found that DEX patients were significantly different and progressed much slower. And they have a couple of reasons that they accounted for this, but in their PATH report, they found, interestingly, a stunning number of patients that were, you know, enrolled right at 2 years. And those 2-year patients actually behave much more like 3-year patients. And, you know, these are not, I may be being provocative, and I'm not sure I would be apologetic about that, but, you know, there was even, there was a question of, were they really at 2 years? Because they certainly didn't behave like that. So there are questions about these data if you look. Now I'm going to move quickly through this because I think there's a published post hoc analysis that we'd be happy to discuss. But one of the things that was part of that paper, and there's been a lot of discussion about creatinine or about biomarkers. This biomarker, and I thought Vincent was going to be here, but this is a paper he and his group published 2005. And I won't read the quote now, but the bottom line is creatinine is a clue, and it's a clue because we know there's impaired creatine metabolism or energy metabolism in patients with ALS. Multiple studies have demonstrated that creatine is increased. Well, how can creatinine be decreased if creatine is increased and there's a non-enzymatic constant degradation? The only reason is because you have impaired ATP production or creatine kinase function. And what we saw in Empower, and we presented these data in Milan, was that the ALS-FRS decline was very well correlated with creatinine loss, and more correlated than with weight loss. There's a disconnect. It's not just as Vincent predicted in 2005, it's not just a passive marker of weight loss. And when you look, actually, interestingly, and there's another discussion about should it be add-on, should it not be add-on. And one of the things that's very interesting is that there's actually a beneficial sparing of creatinine with riluzole alone, with dex alone. But you look at the two together, and there's actually quite a significant sparing of creatinine. And this is more amplified in the LS score definite patients, and especially those with early onset disease. So I think there are plenty of clues about the potential that there may be a ALS phenotype out there that can be biochemically defined. And last but not least, it's not irrational. We still don't know the target of dexpramipexole, but we know a lot of things, and 3 independent labs have shown that it increases ATP synthesis for neurons under stress. And if it increases ATP synthesis, this could account for the increased conversion of creatine to phosphocreatine if that impairment is not because of an enzyme deficiency or absence, but because of a substrate deficiency. And you know what? This is actually measurable with things like MRS. So my last slide is one, and I think this led to what Dr. Schoenfeld told me was going to be a teachable moment when I last presented these data. So I may get a statistical teachable moment, Dan, and I'm ready for that. But, you know, here's neither industry perspective or my perspective, but this is Alvin Feinstein's perspective. And his perspective is that the tragedy of excluding cogent pathophysiologically subgroup analysis merely because they happen to be subgroups will occur if statisticians do not know the distinction and clinicians who do know it remain mute, inarticulate, or intimidated. So I think when you stop looking is really your choice. And, you know, and I don't know that there's a right or wrong answer, but I would say there's more to look for in the Empower study than was just published at one time. So thank you.
So thank you. And that creatinine thing is very interesting because the mice, before they get sick, first indication they start losing weight. And I think the weight measured in humans would not be appropriate because of gastric tube and calorie intake. It would be the lean mass that would correlate with creatinine and kidney function.
BMI didn't correlate either.
The lean body mass. The second thing would be...
Which as I understand it, and this is not my expertise anymore, but you know, the central adipidosity of ALS patients is actually one of the one of the sort of hallmarks that may be indicating the metabolic dysfunction that affects some patients more than others, especially some with systemic disease.
So the second thing would be that your drug, whatever it may be, must not have an effect on the kidney function in preventing the creatinine from being removed. So I think the lean body mass would be the direct measure. If you want to use creatinine as a surrogate, then you have to demonstrate that the kidney function for creatinine clearance is not affected by the drug.
It's not, and I'm not actually here to give any kind of billboard for the pursuit of dexpramipexole. It's actually a stunning eosinophil lowering agent, and we're pursuing that in eosinophil lowering diseases, and we will get back to the hypothesis potentially. I just think there's plenty of data out there in the Empower data that points you to creatinine. I mean, it'd be interesting if Meininger was here, Professor Meininger was here, because I think he would take a different point of view than you about this disconnect between lean body mass. It's not, the creatinine decrease is not related to body mass. It's it's disconnected, and happy to show you the data. It's disconnected in ALS, and you know, but that's it's an opportunity if you choose to pursue it.
So I have a question. So we've been speaking in the last couple of days about enrichment studies, and it seems like what you're really proposing here is an enrichment study to restudy dexpramipexole. Can you explain to us how that enrichment study would work? Which patients you would enrich the study in so you could actually find it?
Yeah, so I've actually had that conversation. There a number of, a handful of investigators here we've had discussions and have actually put in grant applications to do this, and we as a company would certainly be willing to make the drug available under that circumstance. Yeah, I think you should look at short symptom duration patients, less than 12 months, less than maximum 15 months with Ellis-Gorriel definite disease, and I would have them have a creatinine greater than 1 going into the study, because I think that's that's what Vincent saw in 2005. I think that's a clue. And if I were going to do, if we, or when we get the capital, I think you could do an experimental medicine study to test the hypothesis that Dr. Sadiq is questioning about creatine to phosphocreatine conversion and how that relates to creatinine metabolism.
Yoshikazu from Japan, and I have a comment. We completed a study on high-dose methylcobalamin, sponsored by ASI, and that's a phase 2 and 3 study, but that turned out to be 2, and we have to do phase 3. And all the data we collected corresponds to your view. I mean, the shorter duration and Ellis-Gorriel definite was a key for the drug effect.
I think that's the place. That's the place to look.
I'm impressed by your view.
Yeah, thank you.
For our final speaker is Bernard Ravina from Voyager.
Hey, thanks to the organizers for inviting me to talk tonight. I'm not so sure about the 8:30 slot, but we'll make it work. And I'm with a company called Voyager Therapeutics. We are, as Mike was saying, we're small enough that my views on clinical trials are still the company's views on clinical trials. Hopefully that'll continue to be the case for a while. I want to talk a little bit about AAV gene therapy, but really the overall theme of this brief talk is that regardless of modality, regardless of what the mechanism is, you ask the same questions throughout development. How safe is it? Do you get target engagement? Are there pharmacodynamic effects? What's the appropriate dose? Balancing safety and efficacy, and what's the benefit look like? But the way you ask those questions, the designs of the trials and the specific methodologic considerations, I think I'll contend vary extremely with the particular mechanism of action. And so what I say is specific to gene therapy, but I think also applies in good measure to cell-based therapies and other treatments that we think will really have durable effects. So please take it in that light. I want to start off just talking just very generally at a high level about AAV gene therapy. I suspect you'll hear a lot more about this. So AAV or adeno-associated virus has really become sort of the main vector for carrying gene therapies for neurologic disorders. There are a few reasons for that. One is these are not pathogenic viruses. They don't replicate, even non-recombinant, naturally occurring ones don't replicate. They require helper viruses, and when you make them recombinant, you remove all that replication machinery. The other thing that's key is they don't integrate into the genome, right? So they remain episomal outside of the DNA. They're ideal for neurologic disorders for two particular reasons. One, most neurons, at least in the central nervous system, are terminally differentiated. So episomal DNA is defined. It works really well. If you have cells that are turning over, that effect will be lost in the daughter cells. So, and you see these concerns using AAV for, say, liver diseases, for hemophilia, and so forth, where the durability may be an issue. But for neurologic disorders, the durability should be very good, and we have clinical data going out 4 years and animal data, non-human primates, going out 8 years. The other thing is there are concerns about immunogenicity, and if you have high pre-existing titers, you may not be able to get effective treatment with an AAV gene therapy. But since the CNS is relatively immune privileged, antibodies in the CNS are about 1 to 1,000 of what they are in the periphery, it really works very well, especially for something like intrathecal administration. So these AAV vectors, and there's several of them, have really become the workhorse, I think, for a lot of neurologic disorders. This is just a little cartoon of how we think this all occurs. So there are hundreds of naturally occurring AAV serotypes. There are also a lot of engineered serotypes which may evade some of these issues like the immunogenicity. There have been a few of the receptors that have been identified, but basically the virus binds to the cell surface and is endocytosed. It's carrying the therapeutic transgene. It fuses with a lysosome, and there's a pH-dependent activation of phospholipases which allow the virus to escape. How it gets into the nucleus is not entirely clear at this point, but it is clear that it does get into the nucleus and it deposits that therapeutic transgene. Now, this can be used for a lot of purposes. It can replace a missing gene or one that is inadequately expressed, or in the case of SOD1, you can use it to express a small interfering RNA and get knockdown of a toxic messenger RNA. So that's really what we're doing here. So this is very much like an siRNA approach. It's just that you're expressing it in the nucleus. This overall approach has been published by multiple different people. We're working with Bob Brown as well as many others. One of the advantages of expressing it durably is that you use the cell's own machinery to process the RNAs, and that seems to be a little bit safer and less toxic than just dumping shRNAs into the cytoplasm. There are really three working pieces to this overall vector. One is the seed sequence, that's the antisense sequence, to this case in SOD1. Then it's embedded in a microRNA, which really drives how it's expressed and how it's processed, and then what virus, what AAV serotype that you want to package it in. And that all together, especially the serotype, drives how this distributes, what cell types will take it up, and it's really that microRNA in the seed sequence that really drives what kind of knockdown you'll get, and if you'll have off-target knockdown, how specific it'll be. All right, so that is really a permanent or at least long-term approach. So this is really thought to be a one-time treatment, and what I want to do from here is talk about what the clinical trial implications of that mechanism of action are. So it's one-time treatment thought to be due to the immunogenicity, right? So that's related to the capsid. Some people may have pre-existing immunity that may or may not be a problem, depending upon how high it is. But if you give people a bolus of virus, most will, at least for a while, develop a robust enough immune response that you can't retreat. As I mentioned, you do get durable expression, and so you'd expect to have a durable pharmacologic effect unless the cell expressing it dies. And for the time being, these are on the horizon, but they're really not ready for clinical use. The promoters are not regulatable, so we can't turn it up, we can't turn it off either. So what are the clinical trial implications of this? We've really spent the last couple of days talking about a clinical trial paradigm, which is for small molecules, goes 1, 2, 3, and you've talked about what you do in all those steps, but it's really quite different here. So again, the goals, the questions are very similar, but there's fundamentally no single ascending dose study. There certainly isn't any multiple ascending dose study because you're not redosing. You can actually argue that the whole clinical development is one single ascending dose study. We also don't use sort of the dose escalation paradigm that you're accustomed to for either small molecules or biologics. So that's really that sort of start low, you know, several fold below what you think is the active dose, titrate up until you get to the maximum tolerated dose. That's really a paradigm that was driven from oncology, where the notion was that the higher you push a cytotoxic drug, the better a treatment effect you're going to get. Here, you don't want to underdose people because you won't be able to redose. You also really don't want to hit a maximum tolerated dose because you're not going to be able to turn it off. So generally, you look at gene therapy trials, we look at 2 or maybe 3 doses in what we think is the pharmacologically active range. So that's really thinking about it on the safety side of things. On the efficacy side of things, you really have a durable treatment, and it's expressed for several years. So the whole idea here is that unlike a MAD study or a typical phase 2 where you stop treating at some point, you can really use the time dependence to your favor and follow patients such that the first patient treated who will be in the pharmacologically active range could be part of answering that ultimate efficacy question. So the phases of development here really get very blurred and don't hold to, you know, what we're kind of used to when we're talking about small molecules. So 2 implications of that I really want to address. One is this lends itself beautifully to adaptive designs, right? Because you really want to follow these patients seamlessly. The other is it really changes the thinking about when you want to have a control group if you want to use those early patients to ask and answer efficacy questions in a serious way. So I stole this slide from Chris Coffey like 5 years ago, and it still works. And it's just a little schema. This was a course that we put on, and adaptive trials was one of the topics. So when people talk about adaptive trials, it's easy to think that anything goes, and this just nicely outlines that there are a few types of adaptive trial designs. The particular one that is appropriate for this gene or I think cell therapy kind of paradigm is really seamless ones. It's no more appropriate for sort of adaptive randomization or sample size re-estimation than anything else. But the adaptive design paradigm of that first patient being carried through and followed through all the way to the end and asking all those questions about target engagement, safety, and clinical benefit really works very well here. So the next question that I think this whole paradigm really begs is, how do you think about the control group, right? And so the control groups, the whole purpose of a control group is really to address selection bias, right? Patients have different prognoses, you can't compare them. And you've talked over the last couple of days about types of control groups, external or historical control groups, which often really aren't comparable and can only be used in a few situations. And then parallel control groups. And you can use parallel or concurrent control groups. They can be actually matched or they can be randomized. Obviously, randomized is better because you're able to deal with factors that you don't know about that may affect the prognosis. And then it's yet another alternative. In a randomized control group, you can decide to add placebo and double masking. That actually addresses a totally different issue, which is the assessment or rater bias and the biases that the subject may bring to it. So in traditional sort of drug development paradigm with small molecules and antibodies and so forth, how do we usually use control groups? Well, in early studies in SAD and MAD, there usually are a few patients who are randomized and blinded. So you do 6-2 or 3-1 or something like that. They're not really, in that case, they're really being used just for blinding purposes. So when you make your assessments of safety, you don't have any particular expectations about whether they were treated or not. It's not until later on in development that you're really using them as a full comparator group with a statistical comparison of active versus the placebo group. So it's really not until phase 2 or sometimes even until phase 3. In gene therapy, if you want to take advantage of this seamless approach and this long duration effect, you want to start thinking about that actually from the first patients treated. This very much depends on, do you have an administration paradigm that you've got worked out? If you don't know how to deliver the drug, or if you have to work that out, as in some of the cell therapy trials or the gene therapy trials where we're doing direct brain delivery, that obviously you don't want to start off with a control group up front. But if you're simply doing an intrathecal bolus or an intrathecal infusion, like you could do with AAV gene therapy in ALS, you could start with randomized placebo controls up front, and that allows you to use those patients to ask efficacy questions down the road. This is my last slide coming up. So whenever we start talking about placebo control groups, it inevitably begs sort of the ethical questions. And I know some, question whether or not, especially in really small subsets of patients, you should ever be using a placebo control group. But I want to just think with you for a minute about the ethical justification for randomizing patients to a placebo control group. And if you think you're all familiar with this notion of clinical equipoise, this was really an idea that came up as a rescue to preserve and allow people to do randomized phase 3 trials. So Benjamin Friedman developed this in the late 80s in response to concerns that randomizing patients in phase 3 trials after so much had already been learned about a particular drug was not ethical. And they really established this notion that clinical equipoise meant that unless, sort of the broad community of researchers and regulators bought into the fact that the drug worked, there was still broad equipoise, and it would be premature to conclude that it worked, and it was still ethically okay if there was community-based uncertainty to randomize. But the underlying principle here is that earlier on, when there's less certainty and you've asked fewer of the questions about target engagement, safety, pharmacodynamic effects, you have a lot more equipoise. So it actually makes much more sense and is much more consistent with the ethical principles to actually start randomizing early on if you're going to be able to use that information to answer appropriate questions. So I think in this paradigm, you can make the case that the early patients are really entering what can be a long-term efficacy study. There's also an element of real efficiency to this in that you're using that durable treatment effect to answer time-dependent questions. There are also regulatory efficiencies to this as regulatory agencies look at things like breakthrough status, where small amounts of clinical data that are compelling can really speed the regulatory process. And finally, I think viewing this paradigm, which is really kind of the opposite of the typical clinical trials and drug development approach, is in a lot of ways much more patient-centric, right? So you're able to tell the patient community, we don't know a whole lot about this drug or this intervention at this point, so of course it's ethical to randomize, versus we spent years developing this, but still wait, hold on. We've got one last study to do. A lot of people already think it works, but wait a few more years, and hopefully you'll be in the active group. So hopefully I pushed some buttons with this take. If I didn't, I won't feel like I did my job. And I'll wrap up on that. I think there are going to be a number of AAV gene therapies coming down the line. They're exciting. But I think the take-home for this methodologic discussion is really that the mechanism of action drives a lot about how you can and should design these trials. And I would not hold on too strongly to an approach that was developed for an entirely different set of interventions.
So I don't have any industry affiliation, so I can say something I want to say. I think you touched on a very important point, and that is that when we are talking about delivery through viruses, we have a different paradigm. Especially if you're going to raise antibodies, you're actually precluding the placebo group from benefiting from that in the future. And then that's an ethical issue. So either you don't do the study, or you have to develop a different paradigm and convince the regulatory agencies that rules that were previously in place do not apply, and there is compelling evidence that this must go forward.
Yeah. So I think your question begs, what is the placebo group actually composed of? And I didn't address that. So I think it would be a really tough sell to deliver, say, empty viral capsids that would cause an immune response, but you have no potential for any treatment effect. So I think it's much more likely to be a saline infusion or whatever the carrier is for the vector. But I don't think anybody would advocate, and hopefully nobody heard me as saying that, I wouldn't deliver empty capsids. If that would be a phenomenal biological finding if empty capsids worked. We do empty capsids as part of our control groups and cynos and models, but I don't foresee that being part of the clinical development.
So I like very much the approach, and we've been testing a few AAV the past few years in mouse model, and we haven't seen adverse effect. But yet, to go into human, seems to me that the control of gene expression that you want to express should be something that essential. If somehow you express shRNA or a protein that is toxic, you need to turn it off eventually. And I was thinking, what would be the approach? I know the packaging is very small, about 5 kb, 4-5 kb. Is it possible to use 2 different AAV in that case, or manage to go around this, like a tetracycline type?
Yeah, so first let me address the premise of your question, which is a good one. I think there's a lot of human experience with AAV gene therapy with unregulated systems. And it's really been safe. There really haven't been transgene-related issues. So it puts the onus on the preclinical work very heavily, and on modeling and scaling dose appropriately. To your question, though, I mean, the second part, people would really love to have regulatable systems. You're right, there's a limited carrying capacity to AAV. Theoretically, you could deliver more than one AAV, or you could deliver, it could be the same AAV, different batches, somehow, regulated, regulatable elements carried in one, and transgene and standard promoter and others. So, but I do think regulatable systems will come along. They're just not ready for clinic, and they're likely to be leaky even once they're available, I suspect.
Hey, Bernard, I'm excited by this AAV opportunity, certainly since seeing the SMA results this past December, and it's fun to see what you're planning to do with it. The literature on the favorable ethics of early randomization goes back to 72 with Chambers. I can share those papers with you. I'm wondering, another complication I could imagine you're going to encounter is how to do the interim monitoring. You're going to want to make evaluations as you go forward with these, potentially every participant who enters the study or every participant who dies, you might like to be able to make an evaluation of whether you've learned enough to curtail or modify. Have you thought through how you're going to pursue that?
It's a terrific question. It really, it's hard enough running adaptive studies and making decisions when people are blinded. The answer to your question is, have I thought through it? No, I really don't know how we'll run the mechanics like that. But that'll be a big very practical challenge, yeah.
Thanks. So the hour's getting late, and the bar is calling. So I think we're going to skip the panel discussion, if that's okay with everybody. And if you have any specific questions for the speakers tonight, please seek them out. Thanks very much, everybody.